Good afternoon to everyone who are present here physically as well as the ones who have joined virtually through online. We are here today to celebrate the 10th Obaid Siddiqui Memorial Oration to honor the birthday of the father of the modern biology in India, Professor Obaid Siddiqui. As a molecular geneticist, Dr. Siddiqui walked across several different model organisms, starting from bacteria to Aspergillus to Drosophila. He studied and characterized the structure of genes in Aspergillus and later, with Alan Garen, discovered suppressors of nonsense mutations in bacteria. This latter work led to the notion of the stop codons in the genetic code and the mechanism of chain termination during protein synthesis. In the field of neurogenetics, Dr. Siddiqui, along with Seymour Benzer, discovered temperature-sensitive paralytic mutants of Drosophila and the generation and transmission of neuronal signals. He also studied the sense of smell and taste in Drosophila, including how olfactory information gets encoded in the brain of the fly, and how this chemosensory genetics can be studied further. Dr. Siddiqui received several accolades and was a member of several science societies across the world. He was instrumental in setting up the molecular biology unit at TIFR Mumbai and later went on to become the founder director of TIFR NCBS. Apart from that, he also had a great relationship with our founder director, Professor Parthopratim Mojumdar. They met in 1997 at a human evolution conference in the Cold Spring Harbor Laboratory. And interestingly, Professor Siddiqui went there not as a speaker, but as a learner of human evolution, which signifies how much he was passionate about learning new things, even at the age of 65. So like in our interaction today, Professor Lakhotia was also telling that he still learns new things now also. So this signifies that we must learn things until death. So there is no such limit of learning new things. So he was the reviewer of the proposal of the establishment of NIBMG, and eventually he delivered the first Foundation Day lecture of NIBMG on the development of genetics. He succumbed to death from
ঠিক আছে সবাইকে দেখা যাচ্ছে তো
सर्वेषां स्वस्तिर्भवतु सर्वेषां स्वस्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां पूर्णं भवतु सर्वेषां पूर्णं भवतु सर्वेषां मङ्गलं भवतु सर्वेषां मङ्गलं भवतु ॐ सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत्। ॐ शान्ति शान्ति शान्तिः।
Now I would like to invite our director, Dr. Sagar Sengupta, for the welcome remarks and to introduce our today's speaker vividly.
So it is a privilege and with great sense of happiness, I welcome all of you to the 10th Obed Siddiqui Memorial Lecture. And I am very happy for this today, not only because we have a renowned speaker whom I am going to introduce, but also for the fact that this is also my first public interaction after I have joined in IBMG. So as Esha had told that Professor Obed Siddiqui had contributed very significantly to the cause of modern Indian science and modern Indian biology and specifically modern Indian genetics. He is very justly known to be an institution and builder, and we are very happy to celebrate his birth centenary, and we will continue to do that. So for today's talk, I would like to welcome formally Professor Subhash Chandra Lakhotia. And when I thought of names, Professor Lakhotia's name actually came first to my mind, and I immediately called him up, and within a few hours he actually said yes, and I am eternally grateful to that. Professor Lakhotia did his PhD from the University of Calcutta, and he joined for a very short stint as a reader in the Hindu. He joined as a very short stint as a lecturer in the Burdwan University, and then he moved on in a few months' time to Banaras Hindu University in the Department of Zoology. Subsequently, he became the professor in the Department of Zoology again in the BHU, and from 2018 onwards, he is the BHU Distinguished Professor, which he is continuing. Professor Lakhotia's academic and research achievements are many, and if I have to jot down everything, it will take a speech on its own, but I will just give a few salient points about how he has contributed to the study of modern genetics. Professor Lakhotia's doctoral research helped to establish the proposed model of hyperactive male X inactivation to achieve the dosage compensation in Drosophila. Later on, he showed for the first time that heterochromatin in Drosophila is the component which is involved in active transcription. Professor Lakhotia's work actually established also the concept of HNRNPs and how they are involved in regulation of the metabolism. His studies, later studies, also have elucidated a very significant role of the heat shock protein 60, HSP60 in Drosophila, and how they are involved in tracheal development, spermatogenesis, oogenesis, and also in regulating apoptosis, which may have implication in the formation of neoplastic transformation. Professor Lakhotia has published more than 160 research papers, reviews, chapters in books. He has also written many articles on policies of science education, research publication, and ethics in science. Professor Lakhotia has mentored more than 30 PhD students. He has been very actively involved in the improvement of the higher education in science through multiple fora, and he has been a member of the multiple working groups which has been involved in attracting students to science and mathematics. He is actively involved, associated even today with the International Union of Biological Sciences. He is member of his executive company, and later on he was the vice president of that society. Professor Lakhotia has very early in his career, he was the Fulbright Senior Research Fellow, his Senior Scholar Grant, and later on he was conferred with the Shanti Sagar Bhatnagar Award in 1989. He is the fellow of all the 3 national academies in India. He is also a recipient of J.C. Bose Fellowship. And from 29 onwards, he has been the fellow of DST Ramana Fellowship, which I guess is continuing, and also his SERB Distinguished Fellowship. So it is a very long list, and if I go through all the things, it will take a long time, but it has been a wonderful, wonderful privilege and honor to have Professor Lakhotia with us. And now I will welcome you, sir, to please come and deliver your talk.
Okay. Screen share করেছো? Yes, screen share করেছো? Okay, at the outset, I must thank the whole family here for letting me have the opportunity of being in this wonderfully beautiful and exciting place. I'd heard about it. I've known Professor Majumdar for long, and I knew that he's setting up, but I didn't know what kind of setup is this. So it's a remarkable location for me to be here, to see it and to experience it. And I must compliment all of you. And I must thank Professor Sengupta for asking me to come here and give a talk in memory of Professor Ved Siddiqui. I think I first heard him in 1973 when I was at Gujarat University. We had invited him to come and give a talk, and what a talk it was. It was remarkable that in 73, the way he could explain the complex Drosophila, neurogenetics, behavior, and genetic regulation is remarkable. And since then, of course, being in fly community, we have had many interactions. And so it's an honor for me to give a talk in his memory. The topic that I have chosen may look very elementary and in fact, may be very repetitive, but I thought for young students, particularly, it will be useful to go back in history, how the concept of gene has evolved and how it is getting more exciting, more difficult, and more puzzling. And partly, I recall, since I've been teaching since 1971, how my teaching of gene itself has evolved. As things happen, as things develop, and I could sometimes tell my lateral student that what I taught 10 years ago was wrong, because the concept then was that, and then things changed. So I thought I'll just share that experience. This particular picture is very remarkable, and it was, I think it was 1969, that first time it was shown that how genes actually transcribe. It's an EM picture of surface spread chromatin from newt oocyte. And what you see, this Christmas tree continuously are the ribosomal cistrons, multiple copies of them. Is that pointer? Yeah. So what you see here is these are the repetitive ribosomal cistrons, and they're transcribed in a beautiful way. What you see dots, the dots here are the RNA polymerase molecules. And what you see, the strings are the RNA that has been transcribed. So transcription starts from here, and as each polymerase moves, it carries a longer stretch of RNA. There's a beautiful picture which gave a figurative idea of how genes transcribe. And you could see RNA polymerase, you could see the growing RNA, and then at the end, it tends to condense and fold. And this is what it was. So I've been excited since then to look at genes, to see how they are, what they are, and so on. Now, Obaid Siddiqui's academic lineage is remarkable, synthesis of geneticists and physicists. And for that matter, I thought I'll share this picture that the pioneer in Drosophila geneticist, Professor Raichudi, was the first who started genetics after his PhD with Muller. Sometimes he came back after Second World War and set up in Calcutta. And then, of course, his student, Ardina Mukherjee, who finally brought the more recent Drosophila genetics, and where I got trained, and Professor Ashekar Shetty in Mysore, a student of Dobzhansky and Durham, and in Punjab, K.S. Gill, who worked with Strativan. All of them can trace back to Muller. Morgan. This is, and in fact, not only fly geneticists. In 1991, Current Biology published a pedigree. Almost all the leading molecular biologists of the world could trace their academic lineage back to Morgan. That's what Morgan's school had been. But then Siddiqui had a remarkable thing. He did his PhD under Pontecorvo, an Aspergillus, who was Muller's student. Then his postdoc happened to be with Max Delbruck, a quantum physicist turned, whom we call as father of genetics, who had done his postdoc with Strativan, Drosophila geneticist. And so that's where he became geneticist. And his student, Samir Benzer, this is again a very interesting story how Samir Benzer got to do genetics. Those of you who remember R2 locus, that's how he came to do that, bacteriophages. He was a physics student, did his PhD in transistors, and one day happened to listen to a lecture by Max Delbruck, and that changed his life. He decided to go to biology, and in biology, he did the work on the bacteriophages, R2 locus, and later on, I'll come back to this because Benzer has been a remarkable contribution, and Obaid Siddiqui was postdoc with Samir Benzer as well. So that's what a remarkable thing is. So this is a remarkable example of hybrid vigor. You see, you get two, three streams, and you begin to learn things and look at different things. This is what the Pontecorvo and Samir Benzer, and of course, in between the other Drosophila geneticists with whom he worked, and that's what he was doing, all biophysics kind of things. And this is Samir Benzer and Siddiqui discussing something. So that's what remarkable Siddiqui did. Now I come to more about myself. Briefly, what I do now. My research interest happened to be in the non-coding HSR omega gene in Drosophila, in development, thermotolerance, neurodegeneration, and what interactors do this. Then role of stress proteins, particularly heat shock proteins in tumor development and progression. And third area is Ayurvedic biology to find out molecular and cell biological studies on effects of some of these formulations. And of course, as was stated, this is my another interest in higher education research, assessment, policies, and I would say politics that goes on with academics. I don't do it, but I want to fight the politics that goes on. Okay, so now the topic that I'm talking is gene, as I said. Way back in 98, I think, 97, I wrote this article in Resonance, What is a Gene? And because this was something that since I've been teaching, and I thought that students are really confused as to what is a gene. And so I wrote this article. And one of the things that I wrote there was, in the beginning part, that because everybody, newspapers carry genes, so even a layman knows what a gene is. It's no more a very exclusive term. But then the problem is A layman definition of gene is genes determine what we are and that they are made of DNA. This is good for a layman that, okay, they are made of DNA and they determine what we are. What students learn, gene is a segment of DNA of a chromosome that produces a protein. But can we say that? If you ask me to define a gene, I'll raise my hands. I can't define a gene. I can only describe it. I can describe its properties. Why should that be? When a layman can think what a gene is, why should we geneticists worry about it? And that's where I want to talk about how this concept has evolved and how things have changed. Now, this is what has changed with time. So since the time Mendel gave this definition, what he called as factor, which was later renamed as gene. That was the beginning of genetics, we believe. And then maybe after doing all this, if I'm asked to define a gene, I'll go back to Mendel and say it's a factor, it's something that controls a character. But how does it do, how it is organized, that's where the whole things change. And so basically our understanding of gene remains no better than the fabled story of elephants described by a blind man. We all look at gene from different aspects and each one picks up one definition. The layman makes it best. It's the gene that control what we are. But for technicians, for technical things, we need to look at things differently. And we believe that Mendel started it. That's where the classical thing, P experiment breeding, and then Mendels Law, dominance, segregation, event assortment. But remember, Mendel didn't even know that there's a phenomenon like mitosis and meiosis. These phenomena were not discovered. And this is the beauty of Mendel's imagination, visualization, and perhaps his visualizations were far ahead of people around that time to think. And that's why Mendel died a sad person, that he was never recognized in his life. Only post-Mendel life, that it was rediscovered. But then, if you look at history, even before Mendel, there was a person almost 50 years before who had talked about genetic laws of nature. This is something interesting. A sheep breeder, a very rich man, who was doing sheep breeding in Hungary, but out of his experiments, out of his breeding experiments, he thought of these 4 laws. And this is what he called in the paper, the genetische Gesetze der Natur, genetic laws of nature. We think gene term and genetics term was discovered post-Mendel, post-Mendelian rediscovery, that's incorrect. The genetic word, or the German equivalent genetische, was used much earlier. Unfortunately, not many people know. I also learned about this just about 10 months back when one of my former students told me about this. And then I didn't know that there was a paper published in the journal Genetics itself, and earlier in PLOS Biology by these Hungarian scientists who had brought out the history and explained about this. But soon after discovery, we had this chromosomal basis of inheritance. I'll just briefly mention about Sutton and Bovary, 2 very remarkable people. Sutton and Bovary, we learned Sutton Bovary hypothesis, but they never worked together. Actually, they were in different continents. Sutton was a medical student who, during his internship, had done studies with on the chromosomes of grasshopper. And that's where, when he looked at its cytology, the meiotic process, he thought what was Mendel's laws were being seen in grasshopper. So that's where Sutton's discovery was. He was so bright, people thought that he will do PhD and become great scientists, but he actually became a surgeon. And a very good surgeon. Unfortunately, he died young of a disease which he was doing surgery, the gallbladder removal. He was well known for gallbladder surgery, and he himself died of gallbladder surgery. And the history says the day he died, prior to that evening, he was already told by his colleagues not to do anything. He did 4 surgeries because he thought patients are more important than his life. And next day he died. This is remarkable about Sutton. Bovary was another very remarkable person who in 1914 had written an article on cancer. And what he described of cancer is what we are rediscovering. That what cancer is, the mutations, the chromosomal instability, all these are in his 1914 paper. And he was the one who, those who have studied Walter Gilbert's developmental biology book will see that he was the he was the first of experimental embryologists who started experimental embryology and manipulating and discovered remarkable phenomena of chromatin diminution and talked about germ plants, soma plants. So Bovary was a remarkable person. So he also alluded, he never said chromosome basis, but in his writings, he alluded that what happens in meiosis may be what may be happening in. So that's what later on people made it Sutton Bovary hypothesis, but it's not a single hypothesis. And then very rapid developments happen, like Johansson, we typically credit that he gave the term genetics, he gave the term gene, phenotype, and genotype. But as I said, gene is not the term he gave first time. The genetic word was used earlier. And then another person whom I'm sure people here know, Archibald Garrod, one of the first, actually in 1902 itself, he had ascribed that the Mendels factors may be responsible for physiological defects and in what he called inborn errors of metabolism. And then, of course, in 1909, book Inborn Errors of Metabolism was a remarkable book which talked about genetic control of metabolism. And then, of course, the main contribution happened by Thomas and Morgan, who established this famous fly room in Columbia University, and with his students established this thing that gene is the unit of hereditary transmission, function, recombination, and mutation. Now, this is what had been the carrying on definition of gene, that it's one gene is unit of all these functions. And this was based on the gene mapping that had happened, that each gene was mapped by recombination mapping on specific locations. And that's what it was imagined that chromosome can be like a linear line. On there, a point is a given gene, and that's what's called point or a locus. That's how the definition came in. And this was confirmed by subsequent studies on polytene chromosomes, which were discovered in Drosophila. I'll come to that next time, where different genes could be located physically on specific chromosome regions. See, this physical mapping of genes on chromosomes happened in 1930s in Drosophila. In mammalian human chromosomes only when we had the technique of in situ hybridization that we started localized with G-band correlations and so on. But in Drosophila, this was done way back in 1930s. And so what these established, as I said, gene as the unit of function, which controls the phenotype, unit of mutation, generation of new alleles, and unit of recombination between genes. Now, this was important, that at that time, people thought that if there are two genes, the recombination will happen between two genes, not within a gene, because each gene was considered as a point. But this is what was questioned in 1930s. Each gene a single point? Is it divisible? And this question was particularly raised by Muller, that each gene divisible or is it a single unit? And I'm sorry. And so that was one big question, because at that time terms like pseudo alleles came in, that which are behave like alleles but which can recombine. And they may have affect phenotype in a certain different way, and therefore they were called as pseudo alleles. This was a hot concept, but then later on now nobody talks about this concept. If you look at genetics books written in 1970s, you'll find a chapter on pseudo alleles. But now we don't have that, because this term was popular at one time, but then lost its meaning. And then the discovery of polytene chromosomes. You see, this is remarkable. 1934, the paper title itself is Selected Chromosomes and the Attack on the Gene. And this is the last sentence in that paper, last paragraph. With these four discoveries before us, these are hand drawings that Penter had drawn before us, that is constant and distinctive patterns, somatic synapses, the behavior of active and inactive regions, and the separation of the arms of large autosomes. It was clear that we had within our grasp the material of which everyone had been dreaming. We found ourselves out of the woods and upon a plainly marked highway with bypaths stretching in every direction. It was clear that the highway led to the layer of the gene. Notice the word layer. Nobody wants to get into lion's den. But that's where now these chromosomes are allowed. And in fact, this prophetic statement has come out absolutely true, polytene chromosomes. That this is the map drawn by Bridges, C.B. Bridges, and then his son P.N. Bridges. Each chromosome, each line hand drawn. They drew 5,002 lines, each one numbered. And imagine the beauty of their observation. 1930s, no confocal, no high resolution microscope, ordinary light microscope. They drew 5,002 lines. And in 1960s, many people did electron microscopy of these chromosomes to see whether they can see more bands or more interbands. Not one extra, not one missing. Absolutely what he had drawn in 1930s was the number that stays on. And this becomes our guide principle. And now, of course, we have complete genomic correlates. This particular band from this base sequence to this base sequence, it has been all beautifully done. And therefore, this becomes much more than physical map of genes and chromosomes. Then in 1930s, one of the associates of Morgan, Efrusi, and Biddle started experiments using Drosophila. This was the beginning of developmental and biochemical genetics. Pigment synthesis. Why is eye color red? And they found out that there are a number of different pigment molecules which have to come in certain ratio to give the red eye color. They wanted to decide that there were different mutants. Some mutants had 1 million, some had CPI color, some had brown eye color, some had red eye color. So they took each of these mutants, and what they did was developing eyes from Drosophila larva that we call as eye disc, dissected out from one larva of one genotype, injected in another larva of a different genotype, and then let the larva grow and then look at the eye color. And then, and that was the beginning where they said that, what are the question? And so chromosomal pigments have been made from the gene end by the method of genetics and from the character end by biochemical methods. This was the beginning where genes were now no more considered as abstract entities, but as something physical, something that have to do something, function. And this culminated when Biddle collaborated with Tatum and come out with this one gene, one enzyme hypothesis using Neurosporary model, because Neurospora can be grown in minimal medium, and then if you have mutation, one of the enzyme is non-function. So that's what led to the one gene, one enzyme hypothesis. But till now, gene was an abstract identity, a point on a chromosome, a locus, what it is. 40s gave us, before that, another lady, Barbara McClintock, was fighting a very different battle. She realized in May that certain genes don't have a different location on the chromosome map, linkage map. One generation maps on chromosome 9, next generation is somewhere else. And so then she talked of jumping genes or something that can move, saltatory and so on. And the fact is that there was dialogue between two remarkable persons, I forgot the name, which is written in one of the history books, that they had listened to Barbara McClintock's talk. And then after the talk, they were wondering, did you understand what she was saying? And the other said, I don't, but what she says must be true. But nobody knew what it is. It was only in 60s that when in E. coli, they discovered the IS1 sequence, insertion sequence 1, sequenced it and found that in different HFR mutants, they're located in different places, that the idea of what Barbara McClintock was talking in 40s became apparent. And that's where she got Nobel Prize in 1970s. And so we got the transposons or transposable genes or jumping genes as they are called. But this is a discovery that remained inert or ununderstood for long. And today we know that transposons are a remarkable thing. Then in 40s, as I said, gene began to take a material identity. When McClure, McCarthy, and so on, they showed that DNA is genetic material, the RNA genetic material, some viruses. So now we knew that there's a chemical that makes genes. And then finally, two gentlemen sitting outside, Watson and Crick, they defined what it is. And very soon thereafter, okay, one thing that I didn't include in this was Crick became critical in identifying what we call a central dogma. But I should have one more slide. I should have had, which I didn't have. A Russian physicist, George Gamow, an astrophysicist who is supposed to be given an idea of black hole in 1940s. He read Nature paper of double helix, and a theoretical physicist who had nothing to do with biology. He writes a letter to Crick that there must be a code in it, in the DNA structure. And he simply argued that you have 20 amino acids, 4 bases, so each base cannot be one amino acid code. 2 bases, too few amino acids. So there must be 3 bases. because then you can have 64 code words, and these 3 base combinations defines each amino acid. Purely theoretical, but how remarkable. And of course, if you read, there's a very interesting book by Watson, Genes, Gamo, and Girls. That's the name of the book. And of course, it describes Crick's girlfriends and his difficulty in getting married and so on and so forth. But inter alia, it brings out the beautiful story how Gamo argued for RNA to be important. Because Gamo's idea, of course, he thought that DNA becomes RNA, not DNA makes RNA. He thought that 2 strands of DNA, 1 strand goes out, becomes RNA, makes a protein and comes back and becomes DNA. Now, these are very, historically, we may look at what nonsense, but then that's how things have grown up. But he actually, Gamo made it RNA tie club with only 20 members. Each member was given a nickname, one of the amino acids. They will type letters with that name, dear alanine. and this way, and then they will have meeting. His whole purpose was RNA must be made more popular, role of RNA. But unfortunately, that's what subsequent stories will tell us, that this got lost somewhere in the way. Because what Crick had described was, now very often we misunderstand. What Crick had described central dogma only says flow of information, what direction it takes. What it says is it can move from nucleic acid to proteins, but never from protein to nucleic acid. That is the main point of central dogma. Not that DNA makes RNA or RNA makes protein. The information flow is primarily this. Unfortunately, this misinterpretation was Watson's doing. Watson, in his first book on molecular biology of gene, made a statement, DNA makes RNA makes protein. And that's where everybody understood that RNA's only function is to make protein. RNA can do its own independent function. It became a complete misnomer. Every student, each one of us learned central dogma to imagine that DNA has to make RNA, RNA has to make protein, and that's how the information transfer goes on. But for Crick, it was simply the direction of flow of information, how it takes place. And then, of course, one of the major remarkable thing was sickle cell anemia, Ingram's remarkable method of protein fingerprinting. Now, the young students may not imagine what kind of techniques were used, how hazardous techniques were these and how laborious techniques were these. They would digest a protein with trypsin and then separate each peptide, then try to analyze its amino acid composition, and then by combination, permutation combination, describe what the amino acid sequence of the peptide, polypeptide will be. And that's how he came to this idea that there's a mutation in sickle cell anemia. It was single mutation. So this was the first identification where gene sequence was associated with amino acid sequence, DNA sequence. And that's why it was called a point mutation, because that's what geneticists are called. Each gene has a point mutation at a particular locus. And so now it was very clear that one mutation can cause a change in amino acid, and that's what a gene does. That's what they believe. But then in 57, Samuel Benzer, the physicist turned geneticist, remarkable studies on the R2 locus, came out with this final statement that gene is divisible. What we had learned from Morgan's work, that gene is unit of function, unit of mutation, unit of recombination. Now, Benzer could show that all 3 functions are independent. Even a gene is divisible because that's what he calls cistron by cistron's test, and muton and recon. I mean, those who have some poetic ideas can see the rhyming in the names. I mean, scientists are not completely out of literary touch. Cistron, muton, recon. The recombination unit, mutation unit, functional units by cistron's test. And so this was a remarkable thing, that gene is divisible. But then Benzer's contribution is from transistor physics to fast genetics. And remember, where he left R2 locus, it stays there. We don't have much information about the R2 locus function, because everything that he had done, nothing can be done, but there are a lot of things, but I don't think much has been learned. But then in 1961, Benzer moved up. Instead of going down to finer things, he moved to Drosophila, and started chronobiology, behavioral genetics, and then in 91 he moved to neurodegeneration. They built up Drosophila models for all the neurodegenerative diseases, Huntington's, Alzheimer's, Parkinson's, and so on. And this is remarkable. One sometimes wonder, why did Benzer not get a Nobel Prize for his remarkable discovery? But that's what has happened. And then Jacob and Monod came out with this idea that genes can be regulator, can be promoter gene, they can be operator gene, and structural gene, that genes can have different levels of function. Now, for example, the regulator, the promoter and operator are not doing via RNA, by providing some sites for binding of protein. Now, this was a very new concept. And that's where Monod himself made a statement, what is true for E. coli is true for elephant as well. That means what happens in bacteria must happen in higher organs. And C.H. Waddington, a remarkable person who combined genetics and embryology together. In 1930s, geneticists and embryologists won't talk to each other. In fact, some of the great embryologists like Spielman and others, they ask their fellow embryologists not to talk to geneticists because they come out with all kinds of ideas and the genes have nothing to do with embryonic development. But it was Waddington who in 1942 talked of epigenetics, that is during embryonic development, that how different cells take different paths is because of epigenetic modification. And he then also said after the operant model were given, that genes not only act, they can also react because they can respond, they can be regulated. This was a very important statement that he made. And then another important development was Janowski's work that the base sequence in a gene is collinear with amino acid, because what he had shown was that depending on where the mutation is located, now remember, this location mutation was not by sequencing, but simply by genetic mapping, recombination mapping, how frequently this and this map. And then he could show that correspondingly, amino acids get changed in that same linear order. So this was a very important concept of collinearity between gene sequence and amino acid, not gene sequence of bases within a gene and the amino acid sequence in the coded protein. But then came a different discovery, existence of introns. That there are regions within a gene which are transcribed, but then lost. So now the question was, can we talk of collinearity? It was shown that yes, even the exons that are there remain in same order. So collinearity is not affected, but it was Walter Gilbert who suggested that we use the term introns and exons for this a year after their discovery, which was in adenovirus. He suggested that we should call them introns and exons. And then came more complications. We learned one gene, one enzyme, then one cystone, one polypeptide, but now one gene can make numerous kinds of polypeptides by differential splicing. So where is our definition of gene? Can we say one gene, one function? Can we say one gene, one enzyme, one gene, one polypeptide, one cystone, one polypeptide? Now here is a gene that can make, hundreds and different proteins, same gene. And in fact, as I was talking to some students, in Drosophila, there's a gene called DSCAM, which can be a SCAM, but which is a Down syndrome related gene, that's how it is named, can make 35,000 different proteins from same gene by alternative splicing. So now how do we define a gene? Which function? Which phenotype? That's where the complications happen. And then there are more problems. Protozoans throw us numerous kinds of problems. Actually, we often ignore protozoans, but they have very, very remarkable, very different genetic system. One system that has been known for some time is in the beautiful ciliates, like Paramecium slipper animalcule, which has, all ciliates are two kinds of nuclei, one large nucleus, one small nucleus. This large nucleus is a remarkable thing. It's a bag of genes, not long DNA. It had millions of DNA molecules, each molecule of exactly one individual gene. Just one gene, one molecule, not a string of, now how does it originate? Micronucleus doesn't have, it has standard number of chromosomes, standard number of DNA molecules, but then from micronucleus to macronucleus develops something remarkable happens. This DNA keeps on replicating. Then this DNA is cleaved at specific positions, and only 5% of the micronuclear DNA stays in macronucleus. And then each one of them multiplies multiple times. So it's a bag of genes at DNA molecule. This is one problem. But the additional problem is that it has some genes in a scrambled order. Like you see, this is the actin gene. These are 1, 2, 3, that this should be the first part, 2 should be here. This is how the DNA is in the micronucleus. But when it comes to macronucleus, it's beautifully rearranged. They are cut, the thin lines are removed. One is made to join ahead of 2, then 2 joins with 3, 3 joins with 4, 4 with 5, then 5 with 6, and this way it goes on. Now, the question is, who tells the macronucleus what's the order in which these are to be fragmented and joined together? And what we now know is, it's a very simple system. They carry the RNA for this actin gene from the previous generation. And this RNA acts as a template to rearrange them. Now, we neither have now collinearity here. We don't have constancy of DNA. Even immunoglobulin genes in mammals had already questioned that, okay, is there a constancy? We have design variation, but here, and this is not just one, there are multiple such cases. Now, of course, from evolution point of view, one will ask, why should such a senseless organization evolve? We still don't know. But as evolved, it has persisted. All celiacs have this. It's not just Paramecia. It's all celiacs have this kind of organization, scrambled genes. One will have to see whether such things happen in lower animals like sponges or coelentrates or flatworms. We do not know yet. As we discover more and more genomic organization, we will perhaps discover more of such aberrant situations, making life for us more and more complicated. I mean, as I said, when a student asks me what is a gene, I have to say I can't define it, because the moment I say one, immediately exception will come to my mind. And that's a problem. So then now I take a different direction, that parallelly, while genes were being studied, DNA was being studied, parallel developments were happening in cytology, in chromosome work. The two somehow, unfortunately, related remained separate, but then they should have been combined together to understand a better way of understanding genes, what it is. In 1928, a German scientist working with bryophytes, mosses, looking under microscope these chromosomes, and he actually developed this squashing technique, the staining with acetylcholine and squashing and then looking at things. But he looked at these chromosomes in live, sat on microscope for hours together to look at how the nucleus behaved during mitosis. And there he noticed one thing, that some chromatin regions which appeared condensed in interphase remained more condensed in metaphase, and same region next division again remained condensed. While other regions during metaphase where they're condensed, they will open up. So he coined the term heterochromatin and euchromatin. Euchromatin is true chromatin. Heterochromatin is a differential chromatin. This was because of differential staining. So that's what he gave the term heterochromatin and euchromatin. And very quickly, Drosophila geneticists and other geneticists in Mage and others identified that when we map genes, heterochromatic regions don't seem to have any gene map to them. On chromosome region, when you do physical mapping, heterochromatic region don't seem to show any point mutation. So that's where developed the idea that heterochromatin is a gene desert, genetically inert. And this is what the concept was kept on. Even today, we talk of heterochromatin as if it's inactive inert regions. But then in 1960s, people were looking at it. By that time, C-bending was developing, where you could stain specifically heterochromatin with Jigsaw or with fluorescent dyes and so on. So Spencer Brown, who had worked on a very different, unique animal model, coccids, mealybugs, which have a very unusual cycle of development, that males and females both are diploid. But in one system, what happens in males is all the paternal chromosomes are specifically inactivated. For example, here you see that half the chromosomes are dark, half are, and what was shown was that all the heterochromatic chromosomes had come from the father. only maternal chromosomes remain active in males, but in females, both sets are active. And likewise, in mammalian females, one of the X chromosome becomes inactive, other X chromosome remains active, but it's random kind of thing in most eutherians. So that's where he coined this term that heterochromatin can be of two kinds: facultative, which is a developmental regulated process, typically involving only one homolog in condensation, gene inactivation, and constitutive, where both homologs carry same kind of regions inactive. So even heterochromatin now was of two different kinds. Why? It was confusing, but very soon we learned what was. So in 1960s, the heterochromatin was understood that inert or silent, it is differentially condensed, it is variable. Now this was puzzling, that between two related species, you don't have similar amount of heterochromatin. One species may have 80% of chromatin as heterochromatin, another may have only 20%. For example, Drosophila melanogaster has only 20% of its chromatin as heterochromatin. Another species like Drosophila virilis has 80% of its chromatin as heterochromatin. Why this? Why should heterochromatin contain, even in mammals, between different species of mouse or different species of this? We have enormous variation in heterochromatin. This was unexplained. And more important was, heterochromatin was always late replicating in the last part of S phase. But then in 1960s, when the buoyant density DNA suppression happened and in situ hybridization could be done, it was discovered that all heterochromatin regions constituted heterochromatin are rich in highly repetitive and transposable sequences. And they are deemed desert and their transcription is silent, but what evolution biologists learned was that between two related species, what changes is heterochromatin. Euchromatic regions remain same. Heterochromatin is the one, the repetitive sequence. For example, if we take two related species of mouse, what is different in them is the satellite DNA sequences. Now, how does it happen? But what they knew was that heterochromatin or the repetitive sequences are required for speciation. So evolutionary biologists always considered that heterochromatin is important in evolution. How and why that they didn't know, but they knew that functionally it is important. But then, is it really inert? What I had seen in electron microscope was that this is heterochromatic region, and there are typical RNA particles here within heterochromatin. And these were transcriptionally active. This is what I had seen in 1973-74. And so that was the question that we raised. Is it really transcriptionally inactive? Because in Drosophila, it's transcriptionally active. This is one of the first demonstrations that yes, it's active. But then significance remained unknown. But now we know that heterochromatin being inactive, even typical heterochromatin in Drosophila that was supposed to be gene inactive, now we know that numerous genes, both protein coding, transposable elements, non-coding, it's not really poor in genetic sense as the classical genetics has indicated. One may wonder, why did they miss all this? Partly the answer is, we see what we want to see. If I'm convinced that it doesn't have genes, if I see one, no, there must be something wrong. And you don't report it. Now that we do more of DNA sequencing, we can't ignore the genome sequence. We can't look at the transcription that's happening. The other thing that happened was C value, about the haploid DNA content, how much does a species have? It was believed that humans, which we think are the most evolved, we should have maximum DNA. Because we are more evolved, we need more genes, we need more complexity. Maybe our behavior is more complex, but our genome is not. In fact, what is surprising was, insult to human ego, some protozoans had much more DNA than humans have. Single-celled animals. Why should they have? What is it doing there? Of course, one important point was that if we take the minimal DNA content at different levels of organization, it seemed to follow a logarithmic increase. That means, as you become more biologically complex, you need more DNA. But then, why there should be so much of variation? But more important was, which of course human genome confirmed later on, but even earlier, people who had been doing cytology knew that only a small part of this DNA is actually carrying protein coding genes. What is the rest doing? So C-value paradox was typically called paradox. It still remains a paradox, but then we have some answers. We still can't explain why an amoeba needs 100 times more DNA than us. Single cell, which we say most primitive, simplest organism, that has more DNA than us. Why does it have? We still don't have the answer. So because of the light of central dogma, so-called misinterpretation, it was considered that this non-coding DNA is of no consequence, and that's why it's paradox. And that led to this idea of selfish and junk DNA. I often say that if Crick had learned biology, he wouldn't have used the term selfish DNA. See, the approach between a biologist and a physicist. Physicists, chemists will look at a reductionist approach. What is true of one molecule will be true of all molecules. But in biology, one simple thing we say is sum of parts is not equal to whole. Mathematics say 2 plus 2 is always 4. But in biology, you take out components of a cell, put them together, Humpty Dumpty story keeps on. And that's what many of these physicists who came to biology often forgot this, that there are something called organization, something called a system which has to maintain its organization. And so they came out with this selfish DNA. Or what I'm surprised is even Susumu Ono, who was a cytologist and remarkable evolutionary biologist who gave the idea of gene duplication as the basis of new genes evolving, called it junk DNA. Of course, when he said junk, he thought it had a function earlier, but now no more functional. So it's a kind of legacy that organisms carry, but that also didn't explain. And so this, because of this, non-coding DNA remained ignored. In fact, most funding agencies would not fund a project if you are working on a non-coding gene. Why waste money on something which is selfish and of no consequence? They will forget it. And I got trapped in this because very soon after 1980, we are looking at this 93D gene, something that I got interested, was induced by benzamide and by heat shock in all the species that we examined. Genetically, we could find out that yes, it's on the same chromosome, neighbors are same genes, so it's conserved in every species. When we try to look at what protein it makes, it doesn't make any new protein. You induce a gene, but you don't see a new protein. So in 82, we published this paper, and 1980 was given the concept, and everybody agreed it, selfish DNA, junk DNA. But I must say, I never believed in this, that anything can be selfish or junk. I always believed in power of evolution and natural selection, that natural selection will not permit 98% of our DNA to be meaningless, that we are helpless against it, it's just there to maintain itself. As Crick said, selfish DNA. I thought, and learning from evolution biology, that heterochromatin and repetitive sequence are the ones that are most essential, and knowing that in Drosophila, for example, Y chromosome is completely heterochromatic. If it's absent in male, male is completely sterile. So there must be some function of this heterochromatin. And similarly, looking at this gene, it's actively induced this. You see, these are each dot represents an RNA molecule being synthesized. This is tritium labeled and notated gap, so that each dot represents a new synthesis. This was the technique that we use in 1960s, 70s. It was the state of the art technique. Today, it's completely ignored, not known even, because this allowed us to measure the rate of RNA. And I think this is the only method that we can use to measure the rate of transcription. What we can do today is label with BRDU and use immunostaining and fluorescence. Then we can measure how much BRDU, bromuridine has been incorporated, that can give us a rate. But northern PCR only tell us total amount, not the rate. So this was there, and so this was some, and that's where I followed this chromosome. And of course, these beautiful chromosomes, what this shows is, by one of my students had done this, active RNA polymerase staining. And what this dramatically shows is that, okay, there are some regions which are very high, but almost all of the chromosomes you see a reddish stain. Even in this heterochromatic region, you see a lot of red, active RNA polymerase. So obviously, transcription is pervasive. So many regions are being transcribed just meaninglessly. Why should cell spend so much of triphosphates in making RNA? It's not just out of nothing. It must have some function. And that's where I continued with non-coding RNA work. And in 1995, there was a meeting called Trendies, which are started by T. Ram Sharma of ISA Biochemistry. He called them trendies. And these meetings had a very unusual forum. You don't speak about your work. What you think will become trendy in coming years is what you're supposed to speak. I was invited in 1995 at the NIA meeting with Hasnain had organized. And there I talked about that the non-coding RNA are going to be trendy in coming years. And then I wrote a review for my liking for Indian journals. I published in Indian Journal of Biochemistry and Biophysics that RNA polymerase 2 dependent genes that do not code for proteins. And that's what I said, that are no more curiosity or vagaries of biological diversity. They have established themselves as a distinct class of genes with very important functions. And understanding that will be the most important point. And of course, RNA is the first living molecule, so we can't just ignore the first living molecule and throw it away. It can have functions, and that's what we now know. That we have all kinds of names are being given to non-coding RNA, various names. I mean, this can be a very confusing list of names. This is not exclusive. There are still many more names, and many more new names have come out in the last 2 years. In fact, recently, just last week, the Nature Review Molecular Cell Biology carried a review on non-coding RNA principles and how do we nomenclature and future directions. And then what is interesting is, with the complexity, we see an increase in non-coding DNA. As the development becomes more and more complex, we see more and more non-coding DNA. Now, this is not just a coincidence. It must have a meaning. Then what we also know is that RNA plays a very important role in something. This is very exciting from physics point of view, membraneless organelles, phase separated organelles. Like even you see ribosomes, how does it remain together? There's no membrane surrounding it. That's what we call as phase separated, membraneless organelles, or even the whole nucleolus has no membrane, but still stays intact. And so likewise, we have these nuclear speckles, which are just proteins and RNA. In each case, we have non-coding RNA. This is omega speckle that we discovered in our lab on this Atlasar omega 2000, which we call as omega speckles. Again, HNRPs, there's a very unique combination. Protein is in the center, RNA is on the outside. And this has a very particular function because with its different sequences can attach with multiple proteins and thereby can give different meanings. That's where the RNA becomes important. It can attain different structures depending on which protein is bound where. The structure can become different. Even small one single base mutations. See, non-coding RNAs are known to evolve very rapidly. Actually, that was one evidence that they are junk and selfish. This was taken as one evidence that because they evolve rapidly, they can't have function. But now we know that's the function. They can evolve very rapidly and thereby can attain very different functions in different species and can give rise to different morphologies. For example, I give example of our face. Okay, all of us have 2 eyes and 2 nostrils and an upper lip, lower lip. But why does the shape change? We don't have different proteins to make a parrot nose or a sharp nose, a flat nose. Proteins are same. It's the regulation. Which direction, how many times a given cell will divide. And that regulation is what can make a chimpanzee face or a human face or a Caucasian face or a Punjabi face and so on and so forth. And this is what we now need to understand. It's not different proteins, that's a regulation. And that's where these speckles provide a very important function. They trap different proteins. Keep them trapped, release them as when they are wanted. And because RNA is on the outside in most cases, it provides multiple binding, soaking functions, and so on. And then, of course, the more complicated now is transgenerational inheritance. Something that happened in this life can be seen, its effect can be seen in F2, F3 generations also, without genetic change. Epigenetic changes, but what are these? What they are? They can be because of epigenetic modification, chromatin remodeling. They can be because of transgenerational inheritance, paternal. It can be paternal, it can be maternal. Now this is coming up very something very exciting. We are still beginning to understand what it is. Is it because of RNA? Many cases, yes, it's RNA. Do we need to revisit Lamarck's? Because Lamarck said inheritance of acquired characters. This is what it is sometimes showing. Was Lamarck really wrong as we have learned it? Maybe he didn't know, but some observations must have been there. Not the typical, you see, unfortunately, the example of giraffe given in most textbooks and by which students say, okay, Lamarck is wrong, is an exaggeration. I don't think Lamarck had talked in that language, in that way, as the figures are shown. Or the Weizmann's experiments of cutting tail of mouse and then showing that, okay, when after 10 generations mouse comes with tail, that would not be the functional adaptation that Lamarck would have thought. But, you know, in the heat of supporting Darwin's ideas, such experiments were done. And then, unfortunately, some wrong experiments were done. There was a remarkable, I forgot the name of a scientist in late 19th century, who showed that frogs develop the calluses, and he showed images that, okay, because he rubs every time. And then it turned out that this was because he had put ink there and the photograph was showing that calluses are developing. When it was exposed, he committed suicide. But, you know, these kind of examples said, okay, Lamarck doesn't operate anymore. They are completely wrong. But with these examples, we really need to think that can we always talk in terms of natural selection only? This is also, see, actually, in my perception, Lamarck's and Darwin's ideas are not fundamentally different. In fact, when Darwin in 1971 gave the idea of pangenes, this was another term that Darwin had used, pangenes. He had talked to Lamarck's. So from various parts of body, information goes in the germ cell, and that's how the next generation inherits them. So these things we need to really examine. So what I'm okay, I think I should conclude quickly, and I know many of these things for this audience are not new. These are something that you talk every day, you learn every day. But I just thought I'll try to give this message that the diversity of biological systems is enormous, which obviously requires equally diverse information and regulatory circuits. Only a fraction of this is known and understood. No matter how many powerful genomic stations we have and so on and so forth, we still just are beginning to understand what the language is. And a focus only on protein coding genes is grossly inadequate. We need to understand the non-coding component, which is the major component. In humans, for example, 98%, but in some protozoans, it can be, I don't know what percentage of the coding part, maybe only 0.1%, the rest may be all non-coding. Why? Why do they need it that for? And this is what we need to understand the complex regulatory networks that happen. So some of the review that I've written, and this can be a little challenging statement I make. Genomics minus genetics is zero. Just we have sequence, so what? Just we know a child learns alphabets, A, B, C, D, A for apple, B for this, but doesn't know the language yet. We need to learn the grammar, rules of composition, and the meaning. Then only we will understand what genome, what functional components is. And with that, I'll leave you to look at this beauty of chromosomes, which I've fascinated for my life. Fantastic preparation. This, of course, something that I had made 20 years ago for a classroom to show the students how these chromosomes are. And this is after heat shock. And so you see, we can see each gene which is induced by heat shock within 5 minutes. You can see these changes. No other system allows you to look at that. And that's how the whole heat shock response was discovered in 1962 by Retosa, who discovered the ribosomal genes in Drosophila accidentally. He saw his cultures had some unusual puffs, went back to see what had happened, and then he found that the beard incubator temperature has gone up. So then he did an experiment and found, yes, these puffs are. This is the gene of my interest, which is non-coding, and that's also induced strongly. And so we know all this. But this is a beautiful system that can be used for lots of functional genetic studies. So with that, I stop and will be happy to, if questions are there, or if you want questions session to happen.
It's an oration talk.
Okay. All right.
Now I'd like to invite our founder director, the National Science Chair, Professor Parthopratim Mojumdar, to felicitate our today's speaker.
You have to come here. Good. All right.
Beautiful. Very good. Thank you. Thank you. Back it up. That's very good. Thank you. Good. Yeah, yeah. Thank you. Thank you all.
Thank you very much again.
Now I'd like to invite Dr. Kartiki V. Desai on stage for the concluding remarks and the vote of thanks.
Thank you, Isha. And first of all, I would like to thank Professor Lakhotia for coming to our institute, gracing this occasion and taking us through a beautiful, beautiful history of DNA. It also reinforces what a lot of us as teachers are trying to tell the students, and I hope they understand why we are confused when they ask us to define certain terminologies. They sometimes think we are being evasive. So I really thank you for that and coming for this oration. It also gives me great pleasure to say that NIBMJ as a family always comes together when we are organizing talks or orations or any kind of events. And I thank everybody in the administration, because beginning from our senior manager Rajiv ji, Nabarun ji and his team for organizing all the logistics, and Sujata and Seema from the Directorial Secretariat. The reason I began with the administration because they're often faceless and we never see them in the front. Once I've thanked them, and I also thank Vikram and his team, who's our system analyst. He's very successful and is giving a YouTube link live and generally having our talks recorded for us, which we can revisit every time. And now they've thanked the people in the background, I would also like to thank the students who have been on stage and helped us, and also who have attended this talk. And last but not the least, of course, I would like to thank our director, Dr. Sagar Sen Gupta, who thought of inviting you, sir, so we could have this wonderful talk. Our associate directors, Professor Metro and Professor Basu, and all the faculty who have come together to organize this event. And last but not the least, I thank the audience for being present. I know it's a Saturday, but you have honored this occasion. So thank you very much. And with that, I would like to announce that there is a tea organized in the atrium, and we come to the close of the formal program for this day. So thank you all.